right, we are live. So, hello everyone. Thank you for tuning in today for the very first paper talk in ACE's Machine Learning and Neuroscience. Just before we get started, just want to introduce myself. So I'm Robert Boshra. I'm going to be the stream owner for the Machine Learning and Neuroscience. And for newcomers, so welcome to ACE. So this is a community of machine learning practitioners and researchers that are interested in all sorts of machine learning related topics. I do recommend after this talk to just go and watch the other videos. There are videos in all sorts of different topics. And without too much ado, I just want to introduce Dr. Anqi Wu, who's joining us, and I'm very pleased to introduce her as the very first speaker in this series. And she is a postdoctoral research fellow at the Center for Theoretical Neuroscience at the Zuckerman Mind Brain Behavior Institute at Columbia University. She works with Professor Liam Paninsky and Professor John Cunningham. She has received her PhD degree in computational and quantitative neuroscience with Professor Jonathan Pillow from Princeton University in 2019. She holds bachelor's and master's degrees in electrical and computer science from Harbin Institute of Technology in China and University of Southern California. Her research interests lie broadly in Bayesian statistical models of high-dimensional and large-scale neural response and fMRI decoding. She's especially interested in sparse Bayesian structure learning for functional magnetic resonance imaging decoding with hierarchical generative models and Bayesian non-parametric latent variable models for large-scale neural recordings. So the talk today is titled New Methods for Identifying Latent Manifold Structure from Neural Data. And I just want to go ahead and thank Anqi again for joining us today. For the audience, please ask your questions in the chat. I'm going to be keeping an eye out on it and posing questions to Anqi whenever there is time between slides. So with this, I will hand the floor to Anqi, and I'm excited for this.
Okay, great. Thank you so much, Robert, for the nice introduction. Yeah, I'm very honored and excited being invited to give such a to give this talk, which is mainly my my PhD work. So I'm currently at Columbia University, but this is the work that I've done with Professor Jonathan Pillow at Princeton University. So, okay, so the method again is new methods. The talk again is new methods for identifying latent manifold structure from neural data. First, let me present the latent manifold idea. Latent manifold basically means hidden or unobserved manifold. So the question is, so if we ask, can we find continuous low-dimensional structure, high-dimensional sidechain data? Then the latent manifold idea tells us that, yeah, the answer is yes. And there are actually many examples showing that there exists such low-dimensional dynamics underlying multiple cortices in the brain. These dynamics are latent temporal structures. And here is a, oh, actually, can we do the full screen thing? I probably have some animation I want to show. Sorry, Chan, can you, oh, sorry. I just realized this just now. Can we cut it back to the full screen? Hello?
For sure, yeah, we can. I believe we can switch it back to the full screen.
Sorry, because I have some animation that I want to show, and I realized I can't show that in the previous mode. Sorry.
That's all right.
Can people see the screen right now?
Let's just hold on for a couple of seconds to switch to the one.
Yeah, I want to switch it back to the full screen mode. I realized there's an animation that I can't show with this. Oh, okay, sorry. Oh, okay. Can people see my full screen right now?
Yeah, just one window so far.
The full screen? No, no full screen. No, one window. Okay, let me just present share screen. And
Maybe you can just press play just to blow this up to full screen.
yeah, I did play, but then you see one window. I did the entire window. Oh, okay. Sorry. Okay. Can people see that right now? I did
Yep.
the entire screen.
Excellent.
Okay. Really apologize
Right.
for that. I kind of
No worries.
forget. So okay, again, back to the slides. There were many evidence showing in multiple cortices in the brain that there exists a low dimensional temporal structure. So we're convinced that this low dimensional manifold idea would actually work out in for a high dimensional neural population in the brain. Okay. So here is just a motivating example where we have simulated 3 different neurons, which are color-coded in blue, red, and yellow. So here you see each row corresponds to 1 neuron, and the columns are spikes. So neuron usually, the signal of the neural response are usually spikes. Okay, so if you do PCA, the principal component analysis on this small data set, you will see 3 major principal components, which takes basically all of the various underlying the data, and you can't really reduce the dimensionality by throwing away any PC because they all capture a large amount of variance. Here, I'm showing the firing rate, which is the continuous signal generating the spikes. Each color corresponds to a single neuron, which is presented in the first row. So basically, these discrete spike trains are generated from this continuous firing rate signal from Poisson spiking model. So if we plot the firing rate of these 3 neurons in a 3-dimensional neural space, the XYZ coordinates correspond to neuron 1, neuron 2, and neuron 3. And you can see a black moving dot, like moving along a 1-dimensional but curved trajectory. So you can see the black dot moves along the axis 1, like neuron 1, curved, and then come back to the initial point or close to the initial point along neuron 3. So this is saying, although the curve is embedded in a 3-dimensional neural space, but it's actually a 1-dimensional trajectory. So this kind of provides us the insight about why, if you're using a linear dimensionality reduction method like PCA, you see 3 major principal components, but it's actually possible to find a much lower dimension like 1 in this case. So here I'm showing the 2 latent variable, 1-dimensional latent variable X that we use to generate these firing rate data. So X is a 1-dimensional signal. You can think of it's a animal moving, the location of an animal moving in a 1-dimensional linear track. And as the X moves along the time, we can generate the firing rates for each neuron by pumping this latent variable X into the tuning curves that is plotted on the right bottom. So each neuron has a tuning curve function, which is this spiral shape. And then we input the latent location X, and we can generate the firing rate at this corresponding time. So so this is actually the whole generating process we build to generate the spike train at the top of the figure. And we know that there exists a one-dimensional latent trajectory, not the three. So this motivating example just trying to show that using, in many cases, using a linear dimensionality reduction like PCA, you don't see a low dimensional space, but you can use a nonlinear method to find a lower dimensional space. So usually we take a Bayesian approach to do that. In a Bayesian framework, we usually build up two components. One is called the prior over the latent X. The other is the probabilistic mapping from Y to X, where Y is the observed signal, like the neurospectrum data. We only know Y, we don't know X, and we don't know the prior over the latent. We also don't know the probabilistic mapping. But if we build up approach and we estimate the prior and the probabilistic mapping, then given an arbitrary X star on the manifold, we can generate a vector of Poisson fan rates using the probabilistic mapping. So formally, the marginal probability Py, which is the probability of the neural population, is equal to the integral of the multiplication of the observation model Py given X multiplying the prior Px. Okay, so now I'm gonna go into details about how we build this Bayesian model, how we construct the prior over the latent and the probabilistic mapping in a specific scenario where we have population spectrum data. So the model we built is called latent manifold tuning model, where we assume there exists some low dimensional latent process X, which is unobserved. And we have high dimensional neural population spectrums Y, which is observed. Okay, as just mentioned, the first task is we built some prior over the latent P of X. Here, we treat X as a function of time. So we assume the prior for X is X of T is from a Gaussian process prior. So GP here is short for Gaussian process. For people who are not familiar with Gaussian process is some distribution you can use to generate arbitrary functions. And the property of the function would be imposed by the prior covariance K of T. So here, if we assume K of T is a smoothing prior, which means that governing the smoothness of the function X of T, then you sample from such a GP prior, you will get smooth functions. And this smooth function just makes sure that the latent process has the temporal continuity. And then for the probabilistic mapping part, P of Y given X, we assume that, so we basically assume a function called tuning curve function F. So tuning curve is just some function that characterizes the relationship between the neural response with the input latent or observed signal X. So, and usually in neuroscience, tuning curves has to be non-negative. Therefore, we impose a Gaussian process prior over the log of f of X to ensure the non-negativity. Here we have another Gaussian process prior from the first one. This Gaussian process prior is used to impose the continuous, the smoothness over the tuning curve function. And this is biologically plausible because we assume neurons usually fare continuously or smoothly corresponding to the input variable. Like here, in our case, it's the latent location X. And then after constructing such a probabilistic mapping of the P of Y given X, we can generate samples of tuning curves F, and then we can generate the spike rates lambda using the tuning curve for F for each individual neuron. So here, I is the indices for neurons. And finally, given the continuous spike rates, lambda of t, we can generate the Poisson spiking y of t using the Poisson spiking model. So this is the generative model we built. Again, to go through this generative process, at the beginning, we construct a prior P of X using a Gaussian process prior. We sample from such a prior. At an arbitrary time t, we will have X of t. We input this X of t into tuning curve F, which is also sampled from a Gaussian process prior. We can get the spike rate, lambda. And then at time t, given this vector of spike rate lambda, we can get a vector of Poisson spikes y. So this is the generative process from the latent to the observation. But when we really fit the model to the data, we reverse the process. We want to infer the unknown latent process X as well as the unknown tuning curve F from the observed spike train data Y. So then the inference part is to solve the problem. Quickly go through the details about the inference is we first write down the marginal distribution Py as this complicated integral. So Py is the probability of all spike train data. And then it is the integral of the likelihood term, Py given F and X, multiplying the tuning curve prior P of F, which is the GP prior over the log of the tuning curve, as well as the GP prior over the latent process across all the time. And we got this multiplication integrate this over all the unobserved tuning curve as well as the latent process. But this integral is intractable. So we have to come up with some approximate way to solve the inference. The method we propose in this project is called decoupled Laplace approximation, which is used to evaluate the conditional distribution Py given X by integrating over the tuning curves F. If we can get this conditional distribution, even using an approximate way, we can then do map inference for the latent path X hat map by maximizing the joint distribution Py given X multiply the prior Px. So this is the inference is a two-stage thing. We have to iterate between marginalizing over F and maximizing X. So I'm not going to go too much in detail into this inference part, but if people have questions, feel free to ask.
Just a quick question here. So in terms of the data that are actually going in and building that model, these spike trains, are they just time by either firing or not? Like, is it time by binary sort of feature vector over the different neurons that you're recording from?
Yeah, so the observation we have, like the data we have from from the brain is a neuron by time matrix. Each neuron, so each row corresponds to one single neuron, and then each neuron has this spike train data a long time. So the data itself can be binary, which is spikes per se, or it can be discrete value, in which case we can bin the signal, the spike train data, and get the sum of the spike train within a window, in which case it's going to be discrete value, and then you can use Poisson distribution to fit that.
Okay. Very interesting.
Okay, so and then I'm going to talk about some the application of this model. The first I'm still going to show some simulated example from Mobius strip. The interesting thing about Mobius strip is it's a three-dimensional manifold, but there's a twist along the D dimension, so it's different from probably a ball or a cylinder in a three-dimensional space. So here, so in this slide, I'm showing the true manifold, and then we can sample trajectory from such a Mobius strip manifold. On the right, I'm showing the XYZ coordinate for this three-dimensional latent trajectory. And then we can also sample tuning curves from this manifold. So for each individual neuron, and the yellow region means basically you can think of it as a Gaussian bump located on a three-dimensional Mobius strip. The yellow means the high spike rate, and the blue means the low spike rate. So given such fine-tuning curves and the latent we generate in the previous slide, we can generate the Poisson spiking data for 100 neurons. So here again, I'm showing the neuron by time matrix, which is the only observed data we have. And the task is to infer the 3D trajectory and the tuning curves for each individual neuron. So first, again, I'm showing some principal components analysis. If you do PCA over the spectrum data, you will see at least like probably 80 principal components capturing 80 to 90% of the variance in the data, which is certainly not 3. And if you plot the first 3 principal components, here is what we would cover, which nothing like a Mobius strip manifold. But if we can use our latent manifold tuning model, which is an LMT here, to feed the data. And on the top, we show the true manifold. On the bottom, we show the inferred latent plot together, showing kind of where the manifold could be. We can also plot the trajectory along XYZ together with the true trajectory and a linear dynamic system we use to feed the data. So a linear dynamic system basically is we assume the dynamic of the latent follows a linear model, and also the mapping from the latent to the observation is also linear. It's not a bell shape or nonlinear function, it's a linear function. So you can see the XY can do pretty good job, but the Z latent using a linear model, you don't find this highly nonlinear shape. But using our model, we can see the weird like Z dimensional trajectory. So this is showing basically our model can do pretty good job with the simulated Mobius strip example. And then I'm talking about.
Anqi, okay. just before we move ahead, so we just got a question about just where the data are coming from and where these components are coming from. So am I correct in saying that these are sample data that are generated and you made them with 3 latents in mind, like to generate the actual data?
Yeah, so this is simulated, which means we synthetic the data from some underlying model. So so basically we, again, as I mentioned or as I explained in the generative model part, we construct 2 distributions. One is the prior distribution, the other is the probabilistic mapping distribution. From the prior distribution, we can generate the latent trajectory. From the probabilistic mapping distribution, we can generate these tuning curves. And then using these 2 components together, we can generate the Poisson spiking data, but when we try to infer the tuning curve and the latent, we only have this Poisson spiking model, the Poisson spiking data. So we don't have access to the other 2 components.
I see. So let me know in chat if this is clear. Oh, but I also had another follow-up question on. So okay. when you run PCA on this data, you say that it doesn't really, you need a lot of components to capture the variance just because of the nonlinear aspects of the data that you have generated.
Yeah.
Do you know in advance, do you need that a priori knowledge of I have 3 latents in order to run your model, or is that something that you can kind of infer with a completely data-driven approach?
So yeah, that's a good question. For this model, we are not able to automatically identify the number of latent, like 3 in this case. We basically state a 3-dimensional latent model, but when you apply to real data, you have no sense like what the underlying dimensionality should be. So people could use cross-validation, or yeah, you basically vary the number of latent, and you can evaluate some likelihood term to see which is the best number that fits the data or explain the data better. So here I can't really, actually for this model specifically, I can't really automatically learn the number of dimensionality per se.
I see. So this is a method that is really there needs to be more of a field knowledge before you start to apply it, because when it comes to anything in biology, you really need to know, okay, so I'm looking at something that I know from the literature there are 2
Actually,
dimensions per se.
Actually, that's a really good question. So most of the application I have is basically from neuroscience. In neuroscience, people might have some prior knowledge about a data set. Like I can probably talk more about this when okay, what about, let me explain this hippocampal data set, and I'll answer that question. Would that be?
Yeah, sounds good. Thank you.
Okay. Okay. Sorry. Let's hold that question up a little bit, because that's a really good question, especially when it comes to a real like science data set. So here is a rat hippocampus data set that I apply the method to. The data is from Lawrence Frank's lab, and the data set consists of many sessions of recording. The number of the number of neurons vary from 7 to 35. They're all simultaneously recorded. Here I'm showing one example session where they have 35 neurons and 15 minutes recording. And if again we use PCA to find the latent dimensionality, you can see at least we need about 4 to 5 principal components to capture about 60 or 70% of variance in the data. But using our model, like we fit an LMT to the data, but using only 2 latent dimensions, and we can already capture 60% of the variance, which is required by 4 principal components by using PCA. So here I'm showing the model estimate from our LMT. X1, X2 are the 2 dimensions of the latent space, or the latent trajectory. The yellow is our model. By looking at these 2 trajectories, you have no idea what these 2 trajectories doing. But one thing we know that the mouse is traversing in a W-shaped maze when the neurons are recorded in hippocampus cells. So we can plot the 2 trajectory of the mouse while traversing in the W-shaped maze together with our model estimate. But since our model doesn't guarantee, like doesn't have the identifiability to guarantee that we find the exact same trajectory space as the 2 locations, so we have to do some Einstein transformation to align our model estimate on top of with the 2 trajectory, which is the black here. Now you can see our model follows the 2 trajectory of the mouse pretty well. So this provides really nice interpretability of the model estimate by saying that the 2-dimensional latent trajectory of hippocampus place cell are actually monitoring or following the 2 location of the mouse in this W-shaped maze. So this is pretty fascinating and interesting. This is basically saying, by looking at the neural data, we can decode the location of the mouse. When we feed the data, we don't have this location information, right? We only have the spectrum data. And just to interpret the latent we found, we align that with the true location. So back to Robert's question about the dimensionality for a real scientific data set. Here, we know that from previous knowledge in hippocampus place cell study, hippocampus place cells are usually well known for encoding the location of the animal. So, which means presumably you should expect a two-dimensional latent trajectory, right? It's XY coordinate in the spatial location. But what is actually more interesting for us is, or for general neuroscientists, is whether there are other higher dimensional information that are encoded in the hippocampus place cell apart from the 2D locations. So in which case, you might see a 6-dimensional or 5-dimensional latent trajectory. 2 of them encode the 2D location, and the other 3 might encode other information that is not discovered by scientists yet. So, and if we could find out what are the other 3 dimensions are doing, that will provide more scientific value or insight into the place cell in hippocampus. So a short answer is we don't really, I mean, like the prior knowledge in neuroscience, say, would provide some support or some baseline, like provide some support evidence to say, okay, 2-dimensional trajectory is good, but we still want to go to higher dimensions to see whether we can discover something that is uncovered by the neuroscientist. And you can fit, as I just say, you can try 1 dimension, 2-dimensional, 3, up to 6 or 7, fit the data, evaluate them using cross-validation, and try to see which is the best number that explain the data. And we don't really necessarily expect that number to be 2 in this hippocampus place cell case. So hopefully that explains what, Robert's question earlier. And then, okay, so the latent is one thing we are interested in. Another thing is the tuning curve, which is the mapping from the latent to the neural response for each individual neuron. So here I'm showing 4 example neurons. The top row in this left bottom figure is the histogram of the tuning curve by using the true location, and the bottom row is the estimated tuning curve by using the estimated location. We can see we can basically recover, or the estimated tuning curve can match the empirical tuning curve pretty well. So again, so this example, so here is basically a summary of the latent magnitude tuning curve model. We propose a doubly nonlinear latent variable model for spec train data. The model is defined by 2 Gaussian processes. One defines the prior for the latent, the other defines the prior for the tuning curve. And the model is good for extracting low-dimensional nonlinear methods, nonlinear manifolds for place cells. And it can recover, or not recover, it can discover pretty interpretable latent manifolds or latent trajectory, as well as the tuning curve. And they match the empirical data pretty well and provides pretty nice interpretability for the, also pretty nice scientific insight into this hippocampal place cell data. But here is, again, as I just explained, people already have a lot of knowledge about hippocampus. This example is just a proof of concept showing the model can discover something interesting. Next, I'm going to show an application in piriform cortex.
I'll just interrupt here just before we jump into the second application. So I have a question here from the audience. Is the inference analytically solved, perhaps with linked equations and going over iterations, or is there a stochastic approach taken here?
Yeah, so we can't write down an analytical objective function to solve it because of the integral is intractable. So we have to come up with some approximate inference to do that. So we have to use basically iterations of optimization to solve it, not in a stochastic way, but still we do more like a coordinate descent type of thing, and by using approximate inference instead of exact inference with an analytical objective function.
I see. Okay. And so there's also another comment of just so what you were explaining is that basically the domain knowledge can lead into the understanding of like how many latents you could use, but then that could be the feedback loop of actually give more ideas to find new discoveries. And
that's right.
so do you just for the sake of the audience, do you have a can you tell us how noisy the neural data tend to be in the data set that you worked with?
Yeah, sure. The data is pretty hard actually because we so first, like for example, this hippocampal data set, the spike is really sparse. Basically, you have to have a lot of like 10K or even a million time points. Then you can have enough data to discover the latent. And also the number of neurons is very limited. Like science experimentalists won't be able to discover thousands of neurons from the brain. It's only below 100. So when we have such a limited amount of neurons, but also very sparse spike train data, finding the latent itself is a hard task. Using a linear method, usually you don't really do a good job. You have to impose really strong assumptions like regularities, like strong nonlinearity or something to regularize the parameter space so that you can discover something meaningful. Like if you use PCA, I'm not showing the PCA results here, but if you recall the Mobius strip case, if you use PCA, you learn basically nothing, right? The first three principles don't really resemble the manifold, the Mobius strip manifold at all. So you have to really have strong regularities because the data is really, like the SNR is really low in many real neural data sets.
I see. Thank you.
Yeah, no problem. So, okay. Thanks for interrupting here because this is a really good point to clean up some questions or confusions. Again, just to try to transition to the next application. The first application we kind of prove, it's a proof of concept that something people have already had some knowledge about, and we show some results that verifying that prior knowledge. Here, I'm showing an application of the model to the odor responses in piriform cortex in the mouse. So basically, people studying piriform cortex or like animals, basically, how to say, people have not too much, or people study olfactory system or olfactory perception. People have little knowledge about what these olfactory neurons are doing, which is different from visual cortex or hippocampus cells. So our task is to understand, to use neural population in piriform cortex neurons to understand some low dimensional olfactory perception for the mouse when you present different odorants to the mouse. Okay, so the raw data looks like something like this. It's calcium imaging data collected by our collaborator from Bob Data Lab at Harvard University. So they collect olfactory neurons from layer 2 and layer 3 in piriform cortex from the mouse, and then so basically this is a short video. You can see time in the newspaper test the data and reformulate that into a matrix form like this. In the previous example, the matrix is neuron by time. Here, we don't really have the temporal information. We average the neural response across time within a window, and we get a scalar response for this neuron corresponding to a specific order. So here, the raw data is ordered by neuron. Each row is the neural population response corresponding to one odorant. But the idea is basically similar. We still want to find some low-dimensional latent order space. Here I'm showing a two-dimensional case, but it can be arbitrary number. We also want to find the tuning curve, which is the nonlinear mapping function, map the low-dimensional latent to the observed neural response. So the goal of this application is, again, to find the low-dimensional latent order location as well as the neuronal tuning curve using our latent manifold tuning model. A slight difference from the previous example is we don't have the time information. Second, the noise model, the observation noise is not Poisson anymore, it's Gaussian, and it's Gaussian with more structured noise compared with the previous example. So here, I'm gonna quickly go through some technical detail about the new model we're proposing here. It's very similar to the previous one, just with a little bit variant. The first one is for the latent. Previously, we have a Gaussian process prior over the latent with the temporal information. Here, we don't have the temporal, so we only place a normal prior over the location of the latent order. And then, again, for each tuning curve, we still want to maintain the continuity or the smoothness. So we have a Gaussian process prior over the tuning curve of f. Here, we don't have the non-negativity constraints. So it's just f of x, not the log of f of x. And then after generating the tuning curves from the GP prior, we can pick an arbitrary orderant latent location, input that latent location into the tuning curve for each individual neurons, and then we can get the mean response mean response from all the neurons for this specific orderant. And we can repeat this generative procedure for other orderants, and then we can generate the neural population response for all the orderants. But this is only the mean response. This is not the observation. To generate the observation, as we show here, we have to add some noise to the mean response rate. And here, the noise consists of two parts. One is the order-dependent variance. The other is neural correlated Gaussian noise. They multiplying together add to the mean response, and then we generate the observed neural response. I'm not going to go too much detail here, but basically, the main difference you can think of from this latent manifold tuning model from the previous one is, first, we don't have the GP prior for the latent X. Second, the observation noise model is a Gaussian, not a Poisson, and for the noise of the Gaussian noise, it's a noise by multiplying a order-dependent variance with a neural correlated Gaussian noise. Again, the task is to feed this model to the data, and we only have the observed neural response Y, and we want to infer what is the tuning curve F, as well as the latent location axis. The inference is also very similar to the previous one. I'm not going to go too much detail in that. Okay, so here I'm going to show the results that we seek to the real olfactory neural data. Before I dive into the neural response or the latent estimate, let me explain the stimulus a little bit, like how they generate the orderants they used to present to the mouse. So so each orderant can be represented by thousands of chemical features or chemical descriptors, and they map the 1,000 chemical space down to a two-dimensional PC space. And here they pick a set of 22 orderants from this 2D PC space, in which case the first PC corresponding to increasing chain length. Basically, each orderant has, the molecule of this orderant has different carbon chain length. And basically the first PC corresponds to the increasing carbon chain length. and the second PC corresponds to different functional group of these orderants. Here they pick 4 different functional groups: esters, aldehydes, ketones, and acids. Each color corresponds to one functional group, and within a group, the topography of this orderant exhibit this line-shaped structure. Okay, so this is the chemical space, the chemical representation of these orderants. But when they present these orderants, to the animal and collect neural response. For us, neural response
sorry,
signal, we don't have
I
this
just
neural
have
response.
quick interruption.
And also require
quick interruption. Sorry, you were cutting up a little bit. So if you could just reiterate from maybe 20 seconds ago.
20 seconds? I'm not sure it's 20 seconds, but yeah.
So yeah, just a little bit in the beginning of this slide. Thank you.
Okay, so again, first I'm showing the stimulus design where they map the chemical feature space to a 2D PC space. And the first PC corresponds to the increase in carbon chain length of the molecules of the odorants. The second PC corresponds to different functional groups. Within a function group, the odorants exhibit this line-shaped structure, and the carbon chain length increases monotonically with along this line-shaped structure. And then they present these odorants to the animal, collect the neural population response. To feed the model, we don't have access to the chemical representation. Then we can use PCA to find some 2D locations, latent locations of odorants using neural population response only, and you see some structure like showing on the top right there. On the bottom left, we show the PCA. Basically show the accumulated variance using PCA, which is the blue bar, and you can see they require at least 15 principal components to capture 90% of variance of the data. But if we fit LMT model with two-dimensional latent, it can already capture basically the majority of the variance in the data. And also here I'm showing the line-shaped structure, or like the latents we estimate from by using our LMT model. You can see the functional groups are pretty well separated, and within each functional group, the molecules exhibit these, not the molecule, the odorants exhibit this line-shaped structure, and also the orders of these odorants is the same as the order in their chemical space. So even without using the chemical information, we can feed the model and estimate some interpretable latent as shown here. So this really provides some nice interpretability of the 2D space we found. One dimension corresponds to the increase in carbon chain length, and the other dimension corresponds to the functional group. But this 2D information, but these information are unknown by the neuroscientists who study olfactory neural data. So this is kind of an encouraging example showing interesting scientific insight we can find by using our latent manifold tuning model. And then we find these interesting interpretable latent representations for 22 odorants, right? Then what we can do with that? First thing, we can validate for single neuron that whether these latent can generate neural response as what we observe. So remember, we also estimate the tuning curve for each neuron. Here is an example for one example neuron, and we can place the latent of 22 odorants onto this 2D tuning curve to generate the mean response for this specific neuron. The solid bars are the true data we observe, and the light bars are the model prediction, or the model estimate. So we can see the data, the true data matches the model estimate pretty well. So this is saying the 2D tuning curve as well as the 22 odorants the latent representation for these 22 operands can well explain the neural population data well. So this is a good validation. And also we can do prediction for unknown operands. So here I'm showing the prediction performance using a idea similar to cross-validation, but here we call it co-smoothing. But you can think of it as a cross-validation process. How it works is first we estimate the 2D locations for these operands using other neural response except for that one particular neuron. So we can get the so we can get the 2D location for a test order as well in this by using other neural response. And then we place this test order's 2D location to the tuning curve for the selected neuron, and we can generate a predicted mean response for this specific neuron for this unseen test order. And we can see, again, the solid bar is the true data, the light bar is the model estimate. We can see they could match each other pretty well. And that's how we assess the prediction performance by comparing the true data against the model estimate for a specific neuron corresponding to an unseen operand. And then we can calculate the R-square value to evaluate the prediction performance, and we can calculate, we can plot the R-square values of the predicted response to all the held neurons, to all the held orders for all neurons in the population. Here I'm plotting the R-square performance comparison between PCA and our latent manifold tuning model. We can see the red dots are basically above the diagonal, indicating that R-square value of our LMT model are averagely higher than the R-square value using PCA. But again, here, the red dots are actually the latent manifold tuning curve without structure noise, basically with heterogeneous IID noise. By using the structure noise we propose in the model, the performance is further improved. So the conclusion of this slide is the LMT model with structure noise fits or explain the olfactory data well, and it has better prediction performance on how the ordering. So just a quick summary of this LMT with structure noise model. Again, we are able to extract low dimensional olfactory manifold from neural response only. And the low dimensional trajectory, the low dimensional latents are pretty interpretable. We can also identify smooth neural tuning curves over the manifold for each individual neuron. And then finally, we show the model has better predictive performance to head-out orders compared with PCA. Okay, so this is basically the end of the second project. Again, I want to thank for my PhD advisor, Jonathan Polo, and the collaborators on these two projects. So yeah, that's basically the end of the presentation. If people have questions, feel free to ask.
Excellent.
Sure
so
what's the time?
First, thank you very much for a very cool talk. I know that there were a couple of technical issues halfway there, but the talk was phenomenal. I do have one question, and that is actually in regards to the previous project, so the one with place cells.
Okay.
And the question is there a way to physically support the hypothesis of explored place cells? And the follow-up is, can we physically modify those cells after we found them with these methods? So I believe the first part is just about like how well that is supported in the literature and what is typically found there and what has been added in using your models.
Yeah, so for hippocampus place cell, people have already extensively studied it and find, basically they use some simple empirical way, like they calculate the empirical tuning curve by using histograms and to basically explain the neural behavior for each individual neuron. And here I'm just showing that using our tuning curve, we can replicate that. So basically, as long as the tuning curve we estimate is similar to the histogram they found, is a pretty good match. But hopefully we're learning more smooth tuning curve. But there is like more empirical histogram, not a function. So that's the main difference. So although using our model, we resemble what has already been discovered and basically match what has already been studied, but our model is a function, not a histogram. And by learning such a function, you are more flexible to generate whatever you want. So that's basically one part of the motivation. Like, we are not just calculating or just count the number of spikes at different locations. We're learning a smooth function. And for this specific hippocampal data set, the result I'm showing here is using two different dimensions. But I'm interested in exploring higher dimension. What happens in the third dimension? What happened in the fifth dimension, or something like that? That part is still under development and still under exploration. We try to fit to the data and try to interpret the higher dimensional latent trajectory, but we haven't really seen any literature or talked to people yet to see whether those are interesting or those can be helpful for them to study how hippocampal play style or not. So basically, I have to say, hippocampal play style has been widely studied, and it's well known to encode to the locations, but it also encodes some like head orientation, head like speed, velocity, things like that. So those could exist in a higher dimensional latent. We are basically in the process of validating those information. So I can't really answer from that perspective, like how well the previous literature supports the higher dimensional latent. But for 2D, things are pretty well matched and validated.
Right. Well, thank you for a very thorough answer. So I have only one, just because we're running out of time. So just one last question. So how scalable is the model to large data? And just like how does it scale when your number of neurons, I believe in your examples, there were 30 or 100. So what happens if you have collected data from, let's say, 200, 300, 1,000 neurons?
So the number of neurons is not a big issue. The number of the time points is actually a much bigger issue for GP model, because the kernel of the GP, I mean, I have to say we have two kernels. One scales with the number of neurons, but another scales with the time points. But as I just mentioned, in many neural applications, the number of neurons are okay. It's like within 1,000. That's probably the most. So 1,000 by 1,000 matrix is not a big issue. But the time points can increase a lot when you have long session recording, which can be million, in which case we have to deal with a million by million matrix. So there are tricks like inducing points in GP literature allow you to invert such a million by million matrix by using some low rank approximation. So, and the code I developed can scale pretty well. You can basically have arbitrary number of time points you want. So that part has already been addressed, I think, by using some tricks in GP literature. So
very cool.
Yeah.
Okay, so I will ask that one last question because I'm very personally interested in that. Would it work also to have a Bayesian neural network instead of the tuning curves you use in the model?
So that's a good question. So there are two nonlinear components in our model. One is the dynamic, right? We use the GP to model the nonlinear function as a function of time t. Another is nonlinear mapping function from latent to neural response. Especially for the second part, the tuning curve, we use a GP to generate nonlinear function, but there's already literature actually in the field. They use a neural network to construct this nonlinear function to map from the latent to the observation. We also compare to their methods. There are pros and cons. The pros, but for this, especially for this nonlinear tuning curve, we think it's it makes more sense to use GP to generate that, because first we impose smoothness. We want tuning curves to be smooth, right? It fares smoothly from the input space, but a Bayesian neural network or a neural network doesn't really impose the smoothness. So that's one interpretable kind of thing we want to impose to the tuning curve. Second is when you have limited amount of neural data, maybe fitting a neural network doesn't really give you good results. But GP model, like Bayesian model, a GP model doesn't require that many parameters as in a neural network. So it's possible to do a good job when you have limited amount of neural population data. So
I see.
to answer that question is yes, there's people doing that already, but we don't do that way because of the reasons I just explained. Yeah.
I see. Excellent. Well, thank you very much for the answers, and again, Anqi, thank you very much for taking the time to give this talk today. I just want to point everybody to the papers from Anqi. They're a great read, and if you want to dive deep into the work, and also we just had a discussion, and I was told that there's actually code for this model that are available on Anqi's personal website. So feel free to go that, and I'll link it somewhere after the talk. And other than that, I want to thank the audience for some great questions, and we'll come back next month with another talk in the machine learning and neuroscience series.